Hello students, welcome to AriserTalent. As you know, we are studying very important topic like heredity. Okay, so in this lecture I am going to tell you about the Mendel's dihybrid cross. In the previous lecture we have already studied about the Mendel's monohybrid cross. Now we are going to talk about Mendel's dihybrid cross. Mendel's dihybrid cross. What do you understand by dihybrid cross? Dihybrid cross means the cross in which involves two characters, okay, two pair of characteristics, or which characteristics? Contrasting characteristics, okay. So here let me mention dihybrid cross involves two pair of contrasting characters. Okay, so this is the meaning of dihybrid cross, means it involves two contrasting characters of plant, okay, a plant or whatever human being is there. Fine. So here in this lecture we are going to talk about Mendel's dihybrid cross. So what were the features which he chose? That he chose the shape, means round. So first of all the criteria was shape and color. So what are the contrasting characters here? Contrasting characters which Mendel chose for his experiment were round and yellow seeds. Round and yellow seeds. Okay. And the resistant variety was wrinkled and green seeds. Okay, so these were the contrasting characters which Mendel chose for his dihybrid cross. Okay, now let me explain you. We will talk about the dihybrid cross. When Mendel chose the plant which was dominant with the round and yellow seeds. So here the characters were represented by R, R and Y, Y. Capital R means round in shape and capital Y means yellow in color. Okay. So the another which was the dominant variety, sorry, which was the resistant variety, it was wrinkled and green seeds. Wrinkled and green seeds. Okay, so they were represented by, means round and yellow seeds were represented by capital R and capital Y, and wrinkled and green seeds were represented by small r and small y. Okay, when he did the cross pollination between the plant which has round and yellow seeds and another plant which has wrinkled and green seed, then what did he find? He found, what did he found? That in the F1 generation all the plants were round and yellow in color. In F1 generation, he found that all the plants were what? They had round and yellow seeds. Okay. Then when he took the gametes from this F1 generation, what were they? gametes were RY, capital R, capital Y, again capital R, capital Y, okay, then small r, capital Y, and then fourth one was small r, small y, okay. Here small y should be there, capital R, capital Y, okay, capital R, small y, small r, capital Y, and small r and small y, okay. Then when he took the gametes from the F1 generation and then when he allowed them to do self-pollination, okay, what he did, in the when he received all the plants with the round and yellow seeds in the F1 generation, then what he did, he covered the flower, okay, and allowed it for the self-pollination, okay. So what was the result here? We can see here, we can mention capital R, capital Y, okay, capital R with small y, then small r with capital Y, and then small r and small y. Same here, R, capital R, then capital Y, again capital R and small y, then small r, capital Y, and small r and small y, okay. When he allowed the F1 generation plants for the self-pollination, then what was the result here? When it did the self-pollination, means this male gamete did the self-pollination with female gamete, okay. So during the self-pollination, what was the result? Here we can mention the result was capital I. When it was cross-pollinated with this female, okay, this female plant, then capital R and capital Y, capital Y. This was the result. When this gamete was when this gamete was self-pollinated with this gamete, then the result was capital R and capital R, capital Y and small y, okay. Then here we can see capital R, small r, then capital Y and capital Y. Next is capital R, small r, capital Y and small y, okay. Here when this gamete will self-pollinate, will do the process of self-pollination with it, then again capital R and capital Y and small y, okay. Here it is small y and here it is small y, okay. So small y and small y, let me mention small y and small y, okay. Then here we can do the process, sorry, here it is capital, I was right. Here it was capital and then small, okay. Then here self-pollination between these two gametes, capital R, then capital R, capital Y and small y. Both are small y and small y, okay. Then capital R and small r, capital Y and small y, okay. Here capital R, small r, then small y and small y, okay. Then here again capital R, small r, capital Y and capital Y, okay. Then again self-pollination, R, capital Y and small y. Then here small r, capital Y and capital Y. Okay. Then here again R, capital Y and small y. Again here capital R, capital Y, small y. Then capital R and small y, small r, small y and small y. Okay. Then here small r, capital Y and small y. And again small r, small y, small y. So this was the result of F2 generation. Now let me define you what was the result here. Here we can see this was here capital R means the round shape is dominant as well as capital Y is dominant. Okay, so this is the pure plant. We can say this is the pure plant because it contains all the dominant qualities. Okay, so what it will be? It will be round and yellow. Again here it will be round means capital R is dominant. So again it will be round and again Y is dominant. So round and yellow. Okay. Again here R is dominant. Again Y is dominant. So round and yellow. Round and yellow. Okay. Again here capital R and Y are capital Y is dominant. So round and yellow. Next step capital R, capital Y. So round and yellow. Here round and what? Green. Here round and yellow. Here round and green. Okay. Round and yellow. Y is capital, round and yellow. One Y is dominant. Here we can see it is dominant. So it will be round and yellow. Again it will be round and yellow. It will be what? Here we can see it is wrinkled. Wrinkled and here Y is dominant. Capital is here. Okay, so wrinkled and yellow. And here wrinkled and what? Yellow. Wrinkled and yellow. Because here Y is dominant. Round and yellow. Round and yellow. Again round but green. Round and green. Okay. Here wrinkled but yellow. Wrinkled but yellow. Wrinkled and green. Wrinkled and green. Okay, so if we talk about the phenotype means physical appearance. Okay, so according to them, how are round and yellow plants? 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, 9 plants are round and yellow. Okay, so phenotype means appearance. 9. Okay, then how many are round and green? Round and green 1, 2, 3. Okay, round and green are 3. Then wrinkled and yellow wrinkled and yellow are 1, 2, 3. Okay. Then how many are wrinkled and green? Here we can find only one is there, wrinkled and green. So this is the phenotype. Here we can mention this is what? Round and yellow are 9. Okay. And what are they? Round and green. Round and green. These are wrinkled and yellow. And only one is what? One is wrinkled and green. So this is the phenotype of this Mendel's hybrid cross. Okay, this was all about the Mendel's dihybrid cross. Fine. This is the F2 result, F2 generation result. What happened? Mendel took two types of plants, means he chose two characters here. One character was its shape, means shape of the seed, which was round and wrinkled. Okay, and another character was color of the seed. That was, he took one color yellow and another he chose color that was green in the resistant species. Fine. When he took the pollens from the male plant and did the cross pollination with the female plant, then what did he find? That he find in the F1 generation all the plants were, here we can see in the F1 generation all the plants had round and yellow seeds. Okay, then what he did? He covered all the flowers and let them and allowed them to do the self-pollination. In the self-pollination, as a result, he found in the phenotype that 9 plants were round and yellow, 3 plants were round and green, 3 were wrinkled and yellow, means their seeds were wrinkled and yellow, and only 1 plant had wrinkled and green seeds. So this was all about the result of Mendel's dihybrid cross. Okay, now we will talk about the laws of Mendel. Now we will talk about the laws of what? Laws of Mendel. Let's talk about laws of Mendel or Mendel's law. Laws of Mendel. Okay, or Mendel's law. So he gave 3 laws. What were they? Laws of dominance, law of dominance, law of segregation, and law of independent assortment. Okay, so first of all we will talk about law of dominance. What is law of dominance? When the two characters, means when the two characters are taken for the, or we must say when a pair of contrasting characters is chosen for the hybrid, okay, or for the hybrid cross, then only one character is appeared in the F1 generation, and then a character which appears, that is known as dominant character, and one character which does not appear in F1 generation, that is known as resistance. Okay, so let me define law of dominance. First of all we will talk about law of dominance. When a pair of contrasting characters is chosen for hybrid, then only one character appears in F1 generation. This character is known as termed as dominant character. Okay, this is termed as dominant, and the other character which do not appear, the other character which does not appear in F1 generation, is termed as recessive. Okay, so this is the law of dominance. Now we will talk about law of segregation. Actually what happens, that generally alleles or genes remains together, okay, but what they do, they get separate in the time of gamete formation, means they do not get mixed in the hybrid. Okay, so that is the law of segregation. Next is law of segregation. Next is law of segregation. Sorry. What's this? Law of segregation. Okay, according to this law, the alleles or genes remain together. The genes remain together and generally remain together and do not separate or separate at the time of gamete formation. Okay, it means genes do not mix in the hybrid. Okay, means actual genes will remain separate during the hybridization. Okay, next is the law of what? The law of assortment. Okay, independent assortment. According to this law, that the genes, as I told you that the genes remain separate, they do not get mixed in the hybrid in the law of segregation, but in the law of independent assortment, means in the F2 generation, what happens, every gene defines itself properly, or every character remain independent. Okay, so law of segregation, according to law of independent assortment, what happens means the characteristics when two or more pair of contrasting characters, two pair of plants, or we can mention pair of contrasting characters are crossed, then one pair does not get affected with the presence of other. Okay, then the one character does not get affected, or then the inheritance of one, let me mention, then the inheritance of one does not affected by the presence of other. Okay, so this is the law of independent assortment. So in this lecture we studied about the Mendel's dihybrid cross as well as we studied about the Mendel's laws. Hope everything is clear to you. Thank you.